So the second speaker in this lecture series is Dr. Geeta Rai. Dr. Geeta Rai is faculty at Department of Molecular and Human Genetics, BHU. She has done PhD in immunology from SGPGIMS, Lucknow. She has also done postdoctoral fellowship at Laboratory of Immunology, NIH, USA. And she has almost 14 to 18 years of research and teaching experience. She is working in two important fields like immunogenetics and stem cell biology, and she's also working on biosimilar development. And currently, her lab is working on elucidating the genetics of innate immune response in low birth weight infant. She also developing some monoclonal antibodies, and she is working on hematopoietic stem cell and mesenchymal stem cell biology. So with this, I'll request Dr. Geeta Rai to present her talk. Dr. Geeta Rai.
Thank you, sir. I'll just share my slides. Okay, thank you very much to all the organizers, and my greetings to everybody. And I'm very thankful that you all have invited me to be a part of this lecture series organized by your prestigious Society of Inflammation Research. I'm working at Banaras Hindu University, as already introduced by Dr. Mohan Vani. So I'll just go to my next slide and share with you some views of the campus that I am working. This is the campus of Banaras Hindu University. This is its the main gate, that is also known as the Lion Gate. And this is located at the center of the entire campus, the famous Vishwanath Temple. So I'm very happy to have the organizers given me a topic on neutrophils, NETosis, and AIRD, because NETosis is an area which is very fascinating to me, primarily because so little is known in that area, and so much yet needs to be known. So there is a continued interest for picking up research questions. We do know some about the in vitro NETosis. However, about the in vivo NETosis, the factors which regulate it, the mechanisms which are downstream of the entire process, and the different ligands and the stimulations that that induce NETosis do a diverse, they have a very diverse output and manifestation of the entire process. So I'll begin with talking about the neutrophils. So the neutrophils, they are known to be the most important first-line defense molecules. And they are also known as polymorphonuclear neutrophils, primarily because of the shape of the nucleus, the multi-lobe nucleus that it has. And this cell, the neutrophil, is the most abundant leukocyte in the blood, which can account for about 50% of the bloodstream leukocytes. And the lineage-wise, the neutrophils are myeloid in lineage and are characterized by presence of granules and contains enzymes that are potentially toxic to the microbes, invading microbes. Okay. So the granulopoiesis of the neutrophils are that the neutrophils begins, this is the hematopoietic stem cells, which differentiates into the multipotent progenitor MPPs, which then develop into the lymphoid primed multipotent progenitors, which due to the effect of, they develop into granulocyte monocyte progenitors, GMP, which under the effect of granulocyte colony stimulating factor, that is the GCSF, convert into myeloblast. And then through a sequence of polymyelocyte, they convert into myelocyte, then the metamyelocyte, then Ben cells, and eventually become mature neutrophils, and they exit out from the bone marrow into blood circulation. So the neutrophils are very famously known as the surveillance cells because they keep on patrolling your immune system and your body. And the interesting thing about the neutrophils are they're very short-lived with a half-life of only 4 to 10 hours when not activated. And in event of any activation, they immediately succumb. I mean, in the process of fighting to the invaders, they die pretty early. However, the homeostasis is maintained because a large number of neutrophils are produced, that is to the tune of 2 into 10 power minus, to the tune of 11 cells are produced every day, which form the bulk. And in contrast to the blood, the tissue concentrations of neutrophils during the period of homeostasis are quite low. But as soon as there is an invasion of the microbes in the surrounding tissues, there is an influx of neutrophils from the blood, and the number increases. And as I said, the neutrophils are considered as surveillance cells that sweep through the bloodstream, scanning for tissue infections or other inflammatory events, and they can be mobilized from the blood to the sites of infection. So if you, okay. So if you see in this picture, during the bacterial infection, what's happening is that this neutrophil, there are 2 stages that we can call as the rolling of neutrophils. Then there's a step wherein we see the tight anchoring of the neutrophils and diapedesis. So the neutrophils, they are bearing some receptors and ligands like the low affinity state integrins and some glycoproteins, which help them in associating with E-selectins and P-selectins that are present on the endothelial cells of the blood vessel. And then they move through the endothelial cells and come out to reside in the area where the infections are there. That, for example, if you see these are bacteria, so over here they reach. Okay. So what happens when the neutrophils reach the site of inflammation? So when the neutrophils migrate to the sites of inflammation, the first thing that they do is recognize, and using classical mechanisms like phagocytosis or other different cytotoxic mechanism, they would try to engulf and kill the microbe. So once at the sites, the main cytotoxic mechanism that they involve or they are indulged into are phagocytosis. So for example, I'm losing my animation, that's troubling me. Okay, here. So this is phagocytosis is one process. Then they also can degranulate, because we know that these neutrophils have got granules, and these granules, they are released. They release the lytic enzyme in the milieu, and which can neutralize and kill the bacteria. And when the neutrophils are not able to kill the microorganisms using these classical pathways, then there is another very interesting pathway known as the neutrophil extracellular trap, which they use to trap and kill the microbe. So we'll be talking about NETs in further details in the subsequent slides. But in short, I would like to tell you that the process involves decondensation of the chromatin material inside the nucleus. And the decondensed chromatin is then expulsed out from the cell. And in the process of doing so, the nuclear membrane is breached, and also the cell membrane is breached. And this cell undergoes death. So this is also termed as beneficial suicide. And the DNA material or the chromatin material, which is expulsed outside, forms a net-like structure. And in that net-like structure, the microbes are entrapped. Now, along with the DNA that is expulsed out, a lot of antimicrobial peptides are also expunged out. And these antimicrobial peptides, which are released along with these DNA nets, they serve to further neutralize the bacteria. So how does the killing actually take place? So for example, if this neutrophil reaches to the pathogen site, entry site, and there's a phagocytosis or netosis, as the case may be, then it will serve to activate the NADPH, which will result into the ROS, and this ROS will eventually lead to the killing of the microbe. Okay, then the clearance of the neutrophil is also important because in order to maintain the homeostasis. So in the event there are no infections. So once in the tissue, the neutrophils will, after completing its life cycle, will undergo apoptosis. And they're finally cleared through the phagocytes or phagocytes by the resident macrophages and the dendritic cells. In sometimes when the neutrophils, if they don't happen to interact with the invasion, or suppose in their lifetime they do not get to interact with the microbes, then the neutrophils, which naturally age, they start to upregulate the expression of chemokine receptors, CXCR4, and this helps them to return to the bone marrow for final clearance. So this is another way, apart from apoptosis, that the aging neutrophils are removed from the blood circulation. Thus, the clearance of the apoptotic neutrophils is important for controlling the neutrophil production in the bone marrow. And it has been noticed that the phagocytosis of the apoptotic neutrophil has been able to trigger an anti-inflammatory response, which is characterized by reduction in IL-23 by macrophages. And the reduced expression of IL-23 would leads to reduced IL-17 levels and further to less of GCSF production. And the reduction in the GCSF production is again consequential in reducing the granulopoiesis. So these roles that we discussed so far are the primary roles that are attributed to the neutrophil, that is being acting as the first line of defense against the invasion, the innate immunity. But we are also seeing that the neutrophils have multifaceted role, which are now coming up by various research groups, and they have established themselves in different varieties of functions as well. For example, some studies have well established that these neutrophils can also serve as antigen-presenting cells in some situations. They also have a very good interaction with B cells and T cells in the splenic marginal layer, and they have immunosuppressive functions as well. And the neutrophils, which were earlier, it wasn't known that there is a variety of subsets of neutrophils with different attributed functions to it. We will talk about a small subset of the neutrophils in the subsequent slides. And release of nets. So these are some of the functions which earlier were not known, but slowly with the new research coming, we are now knowing that neutrophils can do a lot more than just acting as the first line of defense. So now let's see what NETosis is all about. So in this picture that we have over here, this is just to depict, this is the dying neutrophil. And what we see here is a net or a trap is actually composed of the chromatin material, which has been expelled from the neutrophil. And in green, we see are the microbes, the bacteria. So let's see what neutrophil trap formation is. So this neutrophil trap formation is actually a novel and a unique antimicrobial mechanism, and is also referred to as NETosis. And upon sensing the presence of microbes, as I said earlier, that there is a decondensation of the DNA or the chromatin material takes place within the nucleus, which is then expelled out. And the released traps, as we also call as nets, then are effective in capturing and killing the bacteria and other pathogens from and prevent them from spreading. So this is how activated neutrophil would look like. And this is the microbes which are in the vicinity. So you can see that these are strings and traps which are able to hold and contain. So what are the components of the net? The nets are, the net actually comprises of extracellular webs of DNA, histone proteins, microbicidal proteins, and oxidant enzymes that are released by the neutrophil. Now, if we try to understand what are the sequence of events that take place during the NETosis, if we start from the first trigger, for example, let us consider that this is a neutrophil, and these are receptors which are present on the surface of the neutrophil. So a stimulus which can be a microbe, or actually there's an array of signals which can induce NETosis in neutrophils. But for simplicity's sake, we'll just consider a microbe as the main trigger. So once a microbe triggers the receptor or is sensed by the pathogen recognition receptors present on the cell surface, signals are sent and there's an increase in the calcium, cytosolic calcium level. This increase in the cytosolic calcium level brings about lots of other changes, which include activation of the NADPH oxidase, which will result into the ROS production. These ROS production will lead to the activation of signaling cascades, particularly ERK and P38 or MAPK. This is just one representative pathway. There are other pathways which are also hit. The other thing that will happen will be that some primary granules, which will contain myeloperoxidase enzymes or the neutrophil elastase enzymes, they are also activated, and these release these enzymes, and the enzymes are then translocated within the nucleus. So we also see that by the result of some signal cascades, signaling activation, PAD4, another very important enzyme, which is peptidyl arginine deaminase, which is already there in the neutrophil, is also getting activated. Now we have these three enzymes right in the neutrophil, and each of these three enzymes will work together to act upon these histones by different mechanisms, some of which may be degradation of the histone, or some mechanism could be by inducing citrullination of the histone. The objective of this is to disentangle the chromatin, which is bound by the histone protein. So once this is disentangled, the chromatin material then becomes liberated and it decondenses. The second sequence of that will follow after this will be the breaching of the nuclear membrane. So giving way to the decondensed chromatin to pass out, and also the cytoplasmic membrane is also breached. And so eventually these DNA strands will come out. Now, even initially when the NETosis was explained by Brinkman in 2004 and his colleagues a year later as well, this is what mechanism that we were aware of. But in the due course, it appears that the net release is just not a a simple mechanism following only those processes. It was found that there is a bimodal approach regarding the net release. And the bimodal approach, the decision to pick up one over the other, that is, there isn't enough clarity on to that what dictates that which of the pathway could be followed, although we do have some clue. I'll take you through to understanding what are the different approaches that lead to the net formation. So one of the approach is mediated through the cell death pathway, and the other one does not involve any cell death. In short, that the neutrophils still stay viable after releasing the net through this pathway, whereas the neutrophils lose their integrity and they die, or there's a death which occurs if they form net via this process. So the contrasting points between these two pathways are that in this case where the chromatin decondensation is seen, we don't see much of decondensation here. the nuclear cytoplasmic membrane degradation takes place. And in this case, we do not see any cytoplasmic or nuclear membrane degradation. And expulsion of chromatin into the granular, chromatin and the granule content takes place in the extracellular space, whereas the ejection of DNA is not that widespread. However, this DNA material is expelled out from the cell membrane as vesicles, small vesicles, which are pinched off from the surface. And this pathway, the net formed through this pathway, is referred to as the suicidal NETosis, whereas the net release via this pathway is known as the vital NETosis, primarily because the cells stay vital, they are viable, and they are yet able to perform other processes that they were initially performing. Now, with this broad outline about the suicidal NETosis versus the vital NETosis, let's one more time indulge ourselves into walking through the process of suicidal NETosis, and then I will have the vital NETosis process so that we can do a better comparison for the sake of understanding. So let's begin with the neutrophil. And sorry, let me go back. So this neutrophil, and these are some representative ligands that can serve to activate the neutrophil towards the formation of NET. So as we just noticed in the earlier slides, this ligands will serve to activate and increase in the, lead to the increase in the intracellular calcium levels from the endoplasmic reticulum. And then the sequence of events will lead to increase in ROS production. Also, the ROS would mediate the rupture of granules, which will release the enzymes neutrophil elastase and MPO, and that will translocate to the nucleus. So the neutrophil elastase, the purpose of the neutrophil elastase is to break down the linker histone H1. Just for recapitulation, we know that the histones are there about majorly 5 types of different histones, 2 of which are considered as linker histones, and then there are 3 core histones like H2, A, B, 3, and 4. So these are considered as core histones. It has been noticed that the neutrophil elastase breaks down the linker H1, and the MPO, that is the myeloperoxidase enzyme, augments the DNA chromatin, the chromatin decondensation, also by breaking down the histone. And PAD4, the PAD4 enzyme, this is a peptidyl arginine deaminase. The purpose of the PAD4 enzyme is to modify the arginine and residue in the histone and convert it to citrulline. Now, because the citrulline is not a traditional amino acid, so the histone modification this way is known as the deamination of histone or citrullination of histone. And once these the modification of the histone occurs this way, it no longer is able to hold the DNA coil around it or the chromatin around it. So this will, in a way, disintegrate the assembly of the chromatin material. Now, the last step that will happen after this will be the rupture of the nuclear membrane, and then releasing the free or the cell-free DNA outside, along with the proteolytic, the antimicrobial peptides. Now, after this, let us compare the so this finally would lead to the cell death. And when the cell death will die, if obviously there wouldn't be any mechanisms of physiology that will remain out of this, because it will no more be able to migrate or no more be able to conduct its earlier function like phagocytosis. However, in the case of vital NETosis, the initial triggers could be the same, and the mechanism could also be the same, that is the calcium influx and leading to PAD4 activation. In this case, though, the entire DNA will not be pinched off. We do see that there will be some changes in the shape of the nucleus, but the entire DNA will not be pinched off. Only some part of the DNA will be pinched off from this, loaded into vesicles, and these vesicles will make their way to the cytoplasm, the cell membrane over here, and from the cell membrane they will emit out. So these vesicles filled with NET will come out, and then they are released. So if you look at it microscopically, the way to differentiate a vital NETosis and a suicidal NETosis under a fluorescent microscope would be that in case of suicidal NETosis, you'll see widespread DNA, NET, which can be beautifully stained with different extracellular DNA binding dyes. Whereas in case of vital NETosis, you would see blebs formed on the cell surface. And the interesting thing about vital NETosis is that even after the vesicles filled with NETs are liberated outside in the system, the cell morphology is still intact to a larger degree, and it will still be able to perform its early functions like phagocytosis and chemotaxis, etc. So one more time, let's compare the contrasting points. And there's some important information as well, that what could be inducing one over the other. Like, what are the inducing factors? What are the triggers in the beginning, which will set the decision about whether the suicidal NETosis should be taking place or the viable NETosis. First of all, as a little background about it, it has been shown by different workers at different time points that suicidal NETosis is the more prevalent form of NETosis. And viable NETosis is only a small part of the neutrophils which are involved in the viable NETosis. And the other differences is that it also depends on what is the kind of signal. Sometimes some microbes in particular are able to set off suicidal NETosis, and some different triggers are able to set off viable NETosis. However, still we have to understand the differences or the starting points or trigger points for these two in vivo condition. Now, one question that might come to the mind that if we have to compare NETosis versus apoptosis, because NETosis is also a cell death pathway and apoptosis is also a cell death pathway. So what could be the different contrasting points? How could NETosis be different than apoptosis? Because eventually it is also involved in clearance of the microbes. So the most important thing that I would like to put off is, first of all, the apoptosis is not an inflammatory process. It's actually an anti-inflammatory, whereas the NETosis is highly pro-inflammatory. So the neutrophils, which are highly primed by the cytokines and subsequently exposed to opsonized microbes, take the route of apoptosis. Whereas if there are weaker stimuli, they may lead to NETosis. Now, whether the neutrophils undergo NETosis or they undergo apoptosis becomes a key factor or a key point to dictate the subsequent immune responses. That means whether the inflammation is to set in or the pro-inflammatory scenario will be there. So the NETosis, which tends to be pro-inflammatory and also pro-thrombotic, would lead to release of novel self-antigens. Why I call self-antigens? Because in the subsequent slide, I'll be talking more about it, because as the NETs get released, along with this, a lot of histone proteins get released, normally which was sequestered inside the cell. So because this, our immune system has not been educated against recognizing them as cell. So the double-stranded DNA, the histone proteins, and other nuclear proteins which are now expelled out can be seen as potential autoantigens, and which can trigger off the autoimmune mechanism in the body, and which can evoke the autoimmunity and antibody formation against them. And this is what is primarily happening, because there's a very established connections now which have been drawn between the NETosis and autoimmunity. But before I move on to speak about autoimmunity, I would also like to mention that NETosis is now coming in a big way in case of COVID. And I just tried to look up for some other talk that I was to give some months back. NETosis is now known as the key culprit in the inflammation and the flare that is seen in the COVID. So, and to the extent that the thrombosis or the thrombotic events that are taking place and the cytokine storm that we are talking of, and the widespread inflammation, NETosis is being held as the key stimulating mechanism behind it. And it has become so relevant that a lot of therapeutic drug candidates are being targeted against the components of the NET. I'll try to speak about that in my later slides. Now, because I was trying to link NETosis with autoimmunity, because we know by now that when the DNA material is expelled out, a lot of sequestered antigens they come out in the extracellular milieu, and they give adequate chance for the breakage of the autoimmune balance and the breakage of the tolerance, and autoimmunity is really initiated. So the NET components, as we know, like DNA, histone, neutrophil elastase, cathepsin G, and proteinase 3, other bactericidal peptides like neutrophil defensins, NL37 peptide, etc., which were stored in the granules, they serve as major source of autoantigens in autoimmune diseases, and several autoantibodies to all of these have been reported in different kinds of autoimmune diseases. Another very interesting thing that has been noticed is that the neutrophils, they differentiate into a very different subpopulation. And this different subpopulation is present in parallel with the original population of neutrophils. And this different subpopulation has acquired an ability which is more towards netotizing rather than phagocytizing. One more thing I would like to mention, which I did not mention earlier, a neutrophil which sets into the direction of NET formation would not be able to do phagocytosis, primarily with the fact that the NE and MPO, they tend to disintegrate the actin, rendering it impossible for the NETosis undergoing neutrophil to perform phagocytosis. So the different subpopulation which are noticed in the autoimmune diseases, primarily they are low density granulocytes, which also referred as LDGs. And these LDGs, they represent the pro-inflammatory cytokine, and they have a higher expression of GR1. And the other subset, which has a high expression of CD177, they exhibit anti-inflammatory. So there are two subsets which are being noted in different disease conditions, autoimmune disease conditions. Now, since the topic involved specifically autoimmune rheumatic diseases, so I've tried to include some autoimmune rheumatic disease only in relevance to the NETosis. So what are these? Why do we call them autoimmune rheumatic diseases? It's because that these diseases, they primarily affect the joints, bones, muscles, and connective tissue. And the AIRDs are characterized by an abnormal autoimmune response to normal cells and tissues. They can lead to severe debilitating pain and progressive disease. Some are associated with increased mortality as well. And the abnormal immune response can be directed at a specific part of the body, as we know in case of rheumatoid arthritis, where primarily the joints are involved, or they can be more general and more systemic, like the systemic lupus erythematosus. And diseases like these, like RA, SLE, systemic sclerosis, spondyloarthritis, and systemic vasculitis, they have different clinical presentations, immunopathogenic mechanism. However, they share a common feature of chronic systemic inflammation. And these diseases and some more like these are collectively referred to here as autoimmune rheumatic diseases. Although there are many other rheumatic diseases where the involvement of NETosis had been clear-cut seen. For example, Sjogren's syndrome and polymyositis, they are also diseases which are highly suggestive of involvement of NETosis in the progression. But for the sake of time, I'm only discussing 3 here. I'll be quick because I see the time, I'm running out of time. So the systemic lupus erythematosus, which is characterized by antibodies to the nuclear and cytoplasmic antigen, and there's a multi-system inflammation and the diverse clinical manifestation, which happen to include almost all parts of the body, from the lungs, heart, the kidney, and also manifest as arthritis and skin problems. And so in SLE, we do see a very different subgroup of neutrophil, which are known as the low density granulocytes. And these LDGs have exhibited enhanced capacity to form nets in SLE. And on the right-hand side, what you see here is actually a stained microscopic picture, a microscopic view of an induced net. This is a random picture from my own lab, where I just wanted to show that how an undinduced neutrophil looks like. And this neutrophil, when it is induced by the net-inducing triggers, it will undergo NETosis. And we have stained for markers like DNA staining dye and neutrophil and dyes and citronellitostone dyes. So this is the merge of all the three different stem dyes. So this is how induced net looks like. Now, so this picture that I've taken, I found very nice, and the pictures are also good. This is just to show you as proof that how the LDGs undergo NETosis. This is an example of a study done in SLE. And in the top panel, this is a panel where the neutrophils have been taken from a healthy individual. This one is a neutrophil from a lupus patient neutrophil, a normal neutrophil. And this is the same patient with the different subgroup of neutrophil, which are the low density granulocyte. In the absence of induction, these are just, these cells which are taken, but no induction has been given. Yet, if we stain for NET, we find that the lupus LDGs at the baseline are already in the netoting stage. The lower panel is where these workers have induced them, and then after induction, they have seen that the control cells, that is the neutrophil from healthy individuals, are also now performing NET, because this is very natural. After induction, they will perform NET. So this is just to show that, this is to compare this, that in the absence of induction as well, there was a clear-cut NET in the low density granulocyte, and not so much in the normal neutrophils. This is just another control panel. DMSO is not a great inducer of neutrophils, so NETosis, so we don't see much of NETosis over here. Because the time is running out, I'll also go on and pick up only very important points right now. So this is important though. So the reasons which have been put forward for enhanced NETosis, or the reasons why these NET generated molecule could serve as autoantigen, is there is a clear-cut inefficiency in this patients of lupus to degrade or clear the circulating nets. And because the impaired ability to clear the nets from the system, they happen to stay in the system for much longer than they are required to be. And this gives time for the immune system to recognize them and set up an autoimmune reaction against them. And so I can just move off from this, because these are just examples where in different studies they have been noticed. This one is important, so I'll just put it to you. A cohort of SLE patients was given metformin before giving them a standard care treatment. And it was found that the neutrophils which were pre-treated with the metformin underwent reduced NETosis and extruded fewer mitochondrial DNA copies after PMS stimulation. And this is quite relevant because it has been found that NETosis is also linked to glucose metabolism. So in a way, this also ties up. And the last three things are very important, because I would like to add here that in patients of lupus, researchers have noticed that the DNase activity is highly impaired. Now, what is the importance of DNase activity is the clearance of the NET is done by DNAs. So the net which has come out in the extracellular milieu, they are degraded by the DNases. And if the DNase activity is impaired, then these net degradation will not take place and the clearance will not take place, and they will continue to stay in the system for longer. This is something where we are showing that in case of SLE, this is from my this was from my lab. So we were able to show that as compared to the control, the capacity of the SLE patient's sera was quite impaired for net degradation. And this was when all SLE patients were summed together. But if we tried to group the SLE patients into 3 different groups based on the autoantibody profiles they manifest, it was very interesting to notice that there wasn't much difference in those SLE patients which exhibited only anti-ENA antibodies, but the patients which exhibited anti-DNS and DNA antibodies, they showed much contrasting differences. Now, I'll leave this. This was a summary slide for the SLE, coming to rheumatoid arthritis. And rheumatoid arthritis is a chronic systemic autoimmune disease, which is characterized by synovial inflammation, hyperplasia, and basically involves more of cartilage and bone destruction. And the this is a hallmark that the synovium is involved, and a lot of hyperplasia takes place, and there's a swelling, and the knee joint is really swollen because of the influx of very inflammatory molecules and cytokines in this area. And so this is also a study that I found very interesting, wherein they are trying to show that there was enhanced NETosis in the neutrophil of rheumatoid arthritis patient. And and they were also able to measure that the production of pro-inflammatory cytokines was also stimulated high. Now, I'll also skip this summary, or rather I think I can do a summary, because basically these figures have been drawn by my students, primarily Khushbu. So I'll go through it. So this is a representation of a synovial area, and in the synovial where we have a lot of fibroblasts like synoviocytes, and suppose there is influx of neutrophil, and that neutrophils is showing net production. So this net and the PAD4, which is released from the neutrophil, can serve to activate other neutrophils. And we also see that a lot of cytokines are also secreted over here together, creating a very inflammatory scenario here. So the neutrophils further become activated, and they would also serve to augment the net production. Now, this is an early phase of RA. This is the phase where we can say that the RA has become more established. So we see that a lot of autoantibodies, again, the net components are visible, and these antibodies then bind to the complex, and then they also further exacerbate the disease condition. So we basically talked about the net involvement in rheumatoid arthritis. And before my organizers ask me to stop because I'm already behind my time schedule. So this is the last disease that I will take up, systemic sclerosis. This is a disease which is characterized by limited or diffuse cutaneous sclerosis, microangiopathy, overproduction of autoantibodies to variable organ damage due to vasculopathy or fibrosis. Now, the conditions or the evidence of involvement of NET in this systemic sclerosis is also very significant, because we have the occurrence of oxidative stress in general is linked with the pathogenesis of systemic sclerosis. And interestingly, the PMNs are known to generate important quantities of reactive oxygen species in the course of the disease, and of which the NETosis seems to be an integral part of the pathogenesis, either by promoting tolerance breakdown or by facilitating autoantigen exposure or directly participating in the organ damage. So this is again neutrophils from the patient of, this is a healthy patient neutrophil, and this is a neutrophil from a patient with systemic sclerosis, and which are induced for, which are actually uninduced, and see that without even induction, there's a lot of NET happening from the neutrophils of the patient. And this is the last of the slide that I will discuss with you. This is primarily to show that the study was conducted using PMNs from the SSC patients, which were categorized into 3 groups based on the clinical manifestations. So the 3 groups that were taken were vascular one group had vascular complications, means that the pulmonary arterial hypertension and digital ulcers and scleroderma renal crisis. The second group had fibrotic complications like diffuse phenotype and interstitial lung disease. And the third group, which was taken, was a group with mixed complications, wherein they included patients with at least one vascular and one fibrotic complication. So of these three groups, it was noticed that the patients with had vascular complication had highest level of NETosis ongoing in them. So I think with these studies that I've tried to pull in, it is clear that the neutrophils, the NETosis is very importantly involved in induction of autoimmunity. And so this slide, I just wanted to show that the different NET-associated potential therapeutic targets for which clinical trials are ongoing. And the interesting thing is that these clinical trials which are ongoing are also relevant from the point of view of COVID because their mechanism that they hit upon is common. The way they would control the NET formation will be equally relevant for COVID and for autoimmune diseases and any other diseases which bases upon the dysregulated NET, because there are a lot of other pathology conditions as well, which where the NETosis is underlying in them. So thank you very much. And I would request you all to stay home and stay safe in this very gruesome pandemic.
Thank you, Dr. Geeta. You have given very nice talk and very nice overview on neutrophils, trafficking, their multifaceted role. And you have very nicely presented the suicidal NETosis and vital NETosis and the involvement of NETs in various autoimmune diseases. So now the topic is open for the discussion, question, answer. I request all members to, if you have any questions, please go ahead.
I'm Dr. Chanchekar. I was just wondering, is there any specific marker which help us to figure out that how active the NETosis in a given circumstances?
Yes, sir. The markers which are primarily indicative of increased NETosis happen to be neutrophil elastase and MPO and PAD4. These are found to be very highly upregulated and overexpressed in a scenario where there's a high ongoing NETosis.
We can measure it in the serum or from the local side?
These are measurable in the serum as well. In fact, now the talk is going that, in the COVID, where we normally measure the CRP, IL-6 and dimers, there is a study which has shown the correlation of these enzymes along with these markers which are being measured in the COVID. So these that author which showed these, they suggested that in certain, if you want, you can also use these markers, which are the markers for NETosis, as markers for the disease activity in COVID.
Yeah, because a simple drug like colchicine can block a NETosis for quite a significant extent. Mm-hmm. So that makes sometimes life easy if I can add colchicine in uncontrolled lupus or uncontrolled rheumatoid, if NETosis is the dominant drive there.
Mm-hmm. Yeah, if I go back to the slide, this I did not elaborate much, but the kind of drug targets, the targets that are candidates that are being targeted. One of them is, how you can, use recombinant DNAs, because it has been found that the DNase property, sometimes the DNase happens to be mutated in the autoimmune diseases. So that is not able to render its actual efficiency and effect in clearing. So recombinant DNase can be, administered for stopping or down-regulating the NETosis. Similarly, PAD4 inhibitors and neutrophil elastase inhibitors, they're also in trial because, in fact, one of a company I won't name, for which I'm working as a consultant, I'm conducting a consultancy project, a project in which a molecule which happens to be an inhibitor of PAD4 is being currently investigated, and they aim to do it for lupus. So I'm sure many other labs are involved in, using these net-based targets for therapeutic interventions.
Dr. Deepti has
Dr. Geeta, thank you for the talk. I was just wondering if there's a difference in pathogenesis in these rheumatological disorders, if suicidal is more involved or the vital netosis is more involved. Does it make a difference or?
Yeah, as I said, of the two processes, suicidal is a more prevalent process. The vital process is a very minor process and is not investigated and is not seen ongoing in autoimmunity. I may not be able to tell you that if I have come across enough papers which suggest that vital netosis could be the main players in autoimmunity, but I am confident that there are enough studies which suggest that the suicidal netosis is definitely the key netosis physiology that is going on in these autoimmune diseases.
Thank you.
Dr. Geeta, I have one question, small question.
Yes, sir.
You said that netosis is always a pro-inflammatory effect, right?
Yeah.
But you also said that netosis present antimicrobial peptides. So it's possible to have anti-inflammatory effect also.
Sir, I put it this way. The netosis, the netosis process has to be regulated. If the netosis goes awry or it is dysregulated, it is then it will result into havoc. Because the downside of the netosis is that although the suicide it undergoes is known as a beneficial suicide because it is suiciding itself for the benefit of the host, and it is doing so by clearing of the pathogen, and the inflammatory molecule that it is bringing out is actually to the purpose is to serve the clearance and neutralization of the microbes. Well, this process has to be tightly regulated. In situation, we do see situation where the checkpoint or the regulation of this netosis, when this goes or becomes overwhelming, it becomes disastrous for the body, as the way we see it in the COVID.
Okay. And which is a faster, suicidal netosis versus vital netosis? Which process is fast?
One,
you are saying one is PMA induced.
Actually, why I'm taking time to tell this is because, a lot of contradictory studies. Before, a day before, I came across a paper which said that in 10 minutes of induction, they were able to see vital netosis ongoing, whereas the suicidal netosis takes place all from 15 minutes to 30 minutes. But then there were other papers which I studied earlier which say that the suicidal netosis is something which takes place quickly. So I will be reserved in putting my view on at this point that to suggest which one is faster, but my vote will go to suicidal, because I've learned more from investigators who have suggested suicidal takes place faster. But this is something which I would say that should be, taken as a very reserved answer.
Okay. Any other question?
All right. So I think we are running a race in terms of time.
Yeah.
I just say that she has presented very well. So thank you, Geeta. And also to inform the audience, she also has written a book on netosis. So if anybody is interested, can go ahead and read it. Thank you, Geeta.
Very good. Thank you. Thank you, Dr. Geeta, for a
really
wonderful
wonderful
talk. Yeah.
Thank you, sir. Thank you.